We have probably all seen this type of commercial, a person's testament to why they chose a DNA test. Here we are presented with information that with the Ancestry DNA test, she was able to find out so much information about her family, even finding distant relatives she didn't know she had. Then the commercial ends with something inspirational about discovering your story of your DNA. Here's another ad showing a woman who found her ancestry using 23andMe and having an exciting time traveling to places her ancestors originated. Alluding to the notion that knowing where you came from allows you to find oneself and filling that empty part of who you are. I got to admit, commercials like these make it tempting. Round of applause for the marketing team. The holidays are coming up, and with different sales on DNA tests, I find that I'm almost curious enough to purchase one. But before I purchase anything, I want to answer a few questions that I have. First, I want to know the history of these types of tests. Second, I want to know the science behind them. And lastly, with the multiple different options of DNA tests, what are their advantages and disadvantages, and also what is the accuracy? DNA testing seems to be in the news for various different reasons, from solving old crimes to new benefits of DNA testing. There is also controversy around it, one reason being privacy, wondering who has access to this type of information. But what is the history of DNA testing? What was the initial purpose? Carl Landsteiner in the 1920s discovered the 4 basic blood types that we are familiar with today, A, B, AB, and O, creating the ABO system. With the understanding that blood types are inherited biologically, they can now predict the blood type of a child based on the type the biological parents had. However, the ABO system was too limited to determine definite biological relationships. In the 1930s, serological testing was discovered, and scientists found other proteins on the surface of blood cells. This now allowed a way to identify people, but also couldn't be conclusive for resolving questions about biological relationships, and it was only 40% effective. But the testing is being used in other ways even today. The human leukocyte antigen, or HLA, was discovered in the 1970s, which is a protein present throughout the body with many different types. With the high variability of the HLA types among people, it was used to answer those biological relationship questions. By itself, HLA testing was about 80%. But combined with ABO and serological testing, the power of exclusion was about 90%. It is also important and used in organ transplantation protocols because it determines the likelihood of rejection. A technique called restriction fragment length polymorphism, or RFLP analysis, came out around the 1980s. It was the first genetic test using DNA, which is inherited from biological parents. Compared to HLA and blood proteins, this technique used DNA, and DNA is found in almost every cell, making it even more variable than HLA. This technique was a modern version of science today. This technique allowed scientists to look at a length of DNA fragment and compare it. The idea was that the child's DNA should match their parent, half matching to their father and the other half to their mother. However, some exclusions did apply due to mutations. It also allowed for scientists to look at those who might be at risk for disease and use for fingerprinting and profiling in crime scenes. It's no longer widely used because of the large sample size, which was like the size of 1 pound coin. Also, the time to get the results is pretty lengthy. It could take up to almost a month. Then in the 1990s, polymerase chain reaction, or PCR, was introduced by Carey Mullins. Unlike RFLP, PCR requires a small amount of DNA, like a small cheek swab, and one could get the results within 1 day if needed. The PCR DNA test provided a probability of relationship of about 99.99%. DNA profiling was used for the purpose for crime scenes, paternity testing, and predisposition to diseases. For instance, in Florida in 1987, DNA evidence for multiple rape cases were tied to Tommy Lee Andrews, and he was sentenced to 22 years in prison. He was the first ever American convicted in a case that used DNA evidence. With SNP arrays, or SNP, scientists were able to combine thousands of single nucleotide polymorphism into a single test. This technique is not commonly used for relationship testing, but for a number of other genetic tests like predisposition to genetic diseases, health and wellness, and ancestry, it is also seen as cost efficient. Next-generation sequencing was the newest technique available for relationship analysis that came out in the 2010s. It generates a DNA sequence that is a linear arrangement of letters A, T, C, and G. It is a non-invasive paternity test. Determine the biological father of a fetus as early as 7 weeks gestation, using a blood sample from the mother and a cheek cell sample from the possible father. Safe for pregnancy and detects circulating cell-free DNA in the mother's plasma, and sequences the DNA to interrogate several thousand SNPs. It's now 2020, so what techniques are being done now for DNA testing? And what is the science behind what these different companies are doing? Is it really as simple as to say ordering a test and spitting into a tube, like what is advertised on 23andMe's website? The American Hospital Association projects by 2021, over 100 million Americans will have used a direct-to-consumer take-at-home DNA test. Direct-to-consumer, or DTC, is what we as the general public can purchase from the internet or in store. Tests that are needed to be conducted by one's healthcare professionals are not DTC genetic tests. These types of tests should not be used to diagnose or predict a particular drug or treatment that will work for you. The test requires a small sample of saliva. Ancestry DNA website explains the DNA is processed at a CLIA certified laboratory, usually within 8 weeks, and the results will be reviewed by a physician at the PWN Health. 23andMe runs about almost the same process. It does an at-home based saliva collection kit, and it takes about 3 to 5 weeks to receive results. The standard exams usually include NGS, SNP, Y chromosome, and mitochondrial DNA to look at the different sequences. Mitochondrial DNA, which is contributed by the biological mother, Y chromosome donated by the father, and the markers on the autosomes hold the ancestral information. Modern science makes it possible for you to swab a small cheek sample, and it gives you access to all this new information. In the list of the rigorous standards 23andMe promotes, it states that it provides sub-regional geographical information about ancestry down to the 0.1%. It is also one of the few services that the FDA cleared. So overall, what are the differences between the popular DTC genetic test? I decided to look at 23andMe and Ancestry DNA because those are the two most popular services right now. It was recorded to have about 5 million users each in 2018. Now in 2020, 18 million users database for Ancestry DNA and then 10 million for 23andMe. Almost every DTC doesn't sequence the entire genome, but looks at the points of interest differently. Due to this, results from different companies might come to different conclusions. For instance, Adam Rutherford wrote in Scientific American that 23andMe stated that he did not carry a gene for red hair, while another company said he did, which falls into how the genome is interpreted, with results being confusing or a little misleading. However, both results will give you information about yourself. You might even get the chance to meet a distant relative or just learn more about your ancestors, although new information might not always have that happy fairy tale ending. In regards to accuracy, genetics is a probabilistic science. The accuracy is strongly dependent on the number of ancestry informative markers, or RSMPs, used. More errors occurred with a reduced number of RSMPs, one study found. For an example, scientists looked at East Asian populations, like Chinese and Japanese, and they found that more RSMPs are needed to be used to differentiate. Another look at determining accuracy is your ancestral DNA might not truly represent your ancestors' identity. For instance, let's look at public records. Due to constant changes, which country controls which lands, or countries that are no longer there, your ancestors might not see themselves as what your results might indicate. Your results can only tell you so much. It's not going to be the whole story. My next video, I hope to share the controversy, the advantages and disadvantages regarding genetic testing. Thank you so much for listening.